Okay, all right. Well, listen, thank you very much. I really appreciate the invitation to talk there, and particularly talk about the journey that I've had to get where I am right now, and certainly have lots of time to answer some questions. I will apologize that I'll probably end at around 5 minutes or so before the hour, as I have another meeting I have to get to. But no issues there. This should be an enjoyable hour for both of us, I hope. So I was asked if I would talk a little bit about the journey as a clinician scientist. And so the talk that I've put together has a bit of a mixture of my own personal journey and some insights that I can give you, and also a little bit about our research and what's led us to be thinking along the lines that we are at this time. So what I want to do is we'll just start to go through the slides, which appear. There we go. Okay, so part of what I'm going to do is a little bit of kind of a life history thing and some things that might help you as you're going along, and some quotes from people that I've enjoyed reading or are more anonymous with that. But this one here, grief can take care of itself, but to get the full value of joy, you must have somebody to divide it with. I don't think I'd be where I am right now if it weren't for Wendy. This is my wife. She's been with me since I was an undergrad at Queen's, and we just celebrated 39 years anniversary a week and a bit ago. And it's really been a journey of the two of us. She's actually trained in biochemistry, worked in my lab for a while. And between us, we've really enjoyed this career because we've had a chance to travel, meet a lot of new people, ask questions that I've never been able to imagine that I could before. So it's as much about who you're with as it is where you're going on this journey. And for us, this was also these are our children. So we pass on the values of empathy and kindness to our children by living them. There's no doubt that a career as a clinician scientist and with a small c, big S, spending a lot of time in the lab, that balancing both aspects of that really meant that I wasn't at home as much as one would have anticipated or hoped could occur. And my kids were terrific with this. They've all grown up and have careers and lives of their own now. But striking that balance, I think, will be one of the most challenging things that you'll find. But it is a balance that you need to find because as the days pass and your children grow, you don't get to go back over them again. And so things like camping in the summer times, the kinds of traveling that we did, the one-on-one activities, those types of things become all that more valuable as you move forward through your life. And so I just wanted to say, this is not a journey you take alone. The other piece is that we talk a lot about mentorship. And this is an area of focus at the CIHR, but as a dean and a departmental chair beforehand, we always talked about how we were mentoring, new staff people, the young people that were coming through in our programs. And there's always this sense that there was a singularity of mentorship. And that's actually not true. You will get mentorship throughout your career from a host of people. The individual, sitting here in the middle of the screen for me, this was my supervisor for my postdoctoral fellowship, and that's Ralph Garudo. And I went down to the NIH and worked in the laboratory of Carleton Gajdusek, who had about a dozen years earlier won the Nobel Prize for the discovery of Kuru, which is basically mad cow disease brought on by cannibalism. And so it was a laboratory that was looking at hyperendemic foci of degenerative disease. And I went to work with Ralph. And Ralph was terrific. I actually wasn't sure that I was going to do an academic career. And so I went down for 1 year to see whether I liked it or not. And I stayed for 3. Didn't see any patients at all during that time, just focused entirely on the research aspect of work. But there are other people in this picture, and I won't go through them all. Some of them won't even know that they were mentors. And I think that's the way it should be. They're people that you learn from. They're people who assist you for a period of time of your career, and they change your career. And I remember Ralph often telling me, a really good mentor is the person who influences your outcome, and you don't even know that it's happened. And an interesting way to view it. But this gentleman here, so this is Arthur Hudson, I would say was probably one of the most seminal influences in my life. So Arthur was a neurologist. He passed away a few years ago, sadly passed away with Lou Gehrig's disease. And it was he who really tweaked my interest in studying Lou Gehrig's. And in fact, it was he who really put me in the position of doing a postdoctoral fellowship. I was thinking that I was going to be heading off into private practice. And it was he who one day, actually in front of the nurse's desk, as I was chief resident, kind of said to me, Mike, if you don't go off and do this, you for the rest of your life will regret having passed up the opportunity to think about science and to think about its relationship to the clinical environment in a different way. The other part that was really important about Arthur was that he was ahead of his time in much of his thinking about Lou Gehrig's disease. And I put this up because this is a specific paper which talks about the occurrence of dementia with Lou Gehrig's. And Lou Gehrig's is a motor neuron disease. It was described by Charcot. In fact, it was described by Duchesne and his graduate student, Iran, back around 1830 and some change, as Iran was doing his postdoctoral fellowship. And for over 100 years was believed to be purely a disorder of motor neurons. And as we'll talk about in a moment, that's changed, dramatically has changed in the last decade and a half. But it was Arthur who first recognized this back in the early 80s and did this beautiful paper based only on the literature and our understanding of some of the genomics at the time to describe that, in fact, there could be a florid dementia about 15% of the time. And he was right as the studies moved forward. About the time that I came back, so I came back to Canada in 1990 and started as a young staff person trying to establish my lab at the same time at the Robarts Research Institute and really focused on my ALS component. And there was this slide, but there's a little story behind this slide beforehand. So I'd been back for maybe a couple of years, not much more than that. And I was seeing ALS patient after ALS patient who was having difficulty with emotional control, something we call pseudobulbar affect. And what is meant by that is it's the sudden ability, sudden laughter or tearfulness to an external cue that you would normally bring about a bit of an emotional response, but would never be inappropriate, or it could occur spontaneously, and it can be disabling. People will stop. I've had preachers who wouldn't go up to the pulpit because they might break down for it. I've had people who stopped socializing and so on. Really major thing. And everything that I was trying to do to treat it pharmacologically wasn't working. And one night I was out at dinner. My wife and I were at dinner with a group of colleagues. One of our colleagues here was a neuropsychiatrist actually, Peter Sten. And Peter and I were actually sitting outside, and we remember this day like it was yesterday. We were actually on his deck. I had a scotch. He had a brandy actually. We both had a cigar. These were days in the past. And I turned to him and I said, Peter, I can't treat this. I don't know what's happening. And he kind of looked at me and said, why don't you try these two drugs in combination? So I did, literally next day in clinic. And for the next 20 some odd years, that was the only combination I ever used because it immediately stopped the symptom complex. But the problem is that the receptors on which those drugs should work were not receptors that are found on motor neurons. And it was the first sort of inkling that there was something wrong, that there's something going on that didn't make sense to me as a clinician or as a scientist for that. And then shortly afterwards, I got a call from David Munoz, who was a neuropathologist at Western. And David, who I just loved working with, I was in clinic. I still again remember this one. I was in clinic and he called me up and he said, Mike, you've again made a mistake. Because ultimately the neuropathology is what tells you what's going on. And he said, you got to come down to the morgue. I got to show you this brain because you have gotten it so wrong. And I said, well, no, Dave, I don't think so. Off I went down to the morgue and he showed me this brain. And what he was showing me, sorry, I have a really sensitive mouse, which is going to drive me crazy. What he was showing me was this region of the brain. So the motor strip, precentral gyrus, is located here. It's a little more posterior, right, than most people would think. This is post-central. You're into supplementary motor here. This is anterior cingulate gyrus and hippocampus here of the brain. And so this is mesial frontal lobe here. And just compare it to the mesial occipital parietal lobe here. This is really healthy looking. This is what the normal gyri of the brain should look like. This is grossly atrophic and not at all what you would expect to see in a motor neuron degeneration. And this is what Dave was calling me down to see, because at about that time, we were really starting to understand the frontotemporal dementias. And the word is very much appropriate. When you see this, it's called a frontotemporal lobar degeneration. It shows you that atrophy and was not at all what you would associate with Lou Gehrig's disease. Well, that really led us to start to ask the question about what's going on here. I've now got a pharmacological response that makes no sense whatsoever, and I've got atrophy in a region of the brain that makes no sense. And so we started to prospectively look at a group of patients, 13 of them, in fact, to try and see, could we pick out pathology in this mesial frontal region, and how did it relate? So we used MR spectroscopy, and we looked at the NAACR ratio, so then the accretin and NAACR ratio, and that gives you an index of neuronal health. So as the ratio starts to drop, you're actually losing neurons. And one would expect that here, if we look at the motor cortex, and this group of patients had no evidence that they were actually losing anything in their motor cortex, that precentral gyrus region. And yet, here's a group that were already showing a 50% loss of neurons in the anterior cingulate gyrus, predating the significant motor loss, suggesting that we had a different process going on. And that really started a lot of work. On the one hand, from a clinical side of things, almost 2 decades later, we now have international criteria which describe the frontal lobar degenerations of ALS, which speak to the fact that you can still have pure motor neuron disease. And that's not the primary one. It's less than half the time. The remaining aspect of the time, you'll have some variant of frontotemporal degeneration. It may be a pure frontotemporal dementia in association, in which case, as Arthur had predicted, 15% of the time ALS cases will have that, and 15% of the time frontotemporal dementia patients will have ALS. But there's another roughly 40 to 45% of that will have some variant of it, either behavioral or cognitive or a dysexecutive syndrome. And we now have international criteria, and they're actually called the strong criteria. So after 20 years, we were able to really work internationally to develop these. And they're not just a set of criteria that don't tell us much. They're really meaningful, because if you have evidence of this type of cortical dysfunction in place, this is the Kaplan-Meier survival curve for an individual who doesn't have it. Now, ALS is a fatal disorder, and the vast majority of people will die within 5 to 6 years of symptom onset. But if you have the executive dysfunction, it is a more rapidly progressive disorder. Here's a group that doesn't have it, and so very similar in terms. But so I want you to remember this, because we're going to come back to this towards the end of the talk. So there is a significant impact of having this dysfunction on the disease itself. And there's been a whole host of reasons for it, but I'm going to show you what we think to be the reason later on. Now, this is where it got really interesting, because that same group of patients that we were studying initially by the MR spectroscopy came to autopsy. And we began to look at their brains, and particularly, for the obvious reasons, at the frontal neocortex, because this is where we saw the evidence of neuronal loss. And so we're hearing, or you're seeing here, frontal cortex, where in fact, this is very healthy. And here, you don't even need a microscope. We could hold these slides up to a light bulb, and you could see that you have this moth-eaten appearance here at the junction of really cortical layer 2 and cortical layer 3. And if you were to see this at any other time, you'd think that this looks like, well, it is a superficial linear spongiosis, but you'd think of this as a prion-based disorder. And yet we did a whole lot of work trying to figure this out, and we couldn't, until we get a bit of a surprise. And this was one final bit of studying analysis that we did on that particular group. And it was led by an individual who came to us as actually as an orthopedic trauma surgeon from China, who was looking at doing some postdoctoral work, and who had had his undergraduate work was in neuronal tracking of occipital cortex during development. And he had used tau tracking to do it. So we asked him to look at that. And that led us on to a pathway of work that this individual here, Wencheng Yang, really spearheaded for us. And this would be my lab around the late 1990s, early 2000s. And Wencheng, as I say, was a trauma surgeon who had come across and wanted to do this work. And it was really he who first identified that there was this deposition of tau proteins in neurons that we had never seen there before with that. Now, tau is a phosphoprotein. It's really important in terms of axonal transport. It helps to stabilize the microtubule system. Phosphorylation is a normal event in the life history of it, but you can have pathological phosphorylation either by too many phosphate residues, in which case you're taking a negative charge and putting into play on this, which will open up what we know to be a bit of a paperclip structure. So it kind of holds in on itself, and when you start to phosphorylate it, it opens up, and that changes interactions of specific domains. And I'll come back to that. So we wondered, could it be that this is pathologically phosphorylated in any way? And to make a long story short, the answer was obviously yes. Otherwise, I wouldn't be giving this talk because we hadn't gotten anywhere. But the answer was that we actually found that there were specific residues, and particularly threonine 175, which appeared to be uniquely phosphorylated in the ALS population, both those that were cognitively impaired and those that were not. And we'll come back to that differentiation in a moment. So we now had an epitope that seemed to make some sense. So what Wencheng did was he then worked to create an antibody against that epitope. And this antibody, which specifically looks at phosphorylation at the 175 epitope, he then did an amazing study to look across ALS patients, those that we knew were cognitively impaired and those that were not. And again, this is frontal cortex, but we found it in several other areas. And you can see this deposition of tau protein, sometimes as pathological fibril-like structures within neurons, sometimes as dystrophic neurites, sometimes as what we would call otherwise argentophilic granules, if this was a silver stain. And if I showed you this, you'd begin to say, well, isn't this just Alzheimer's disease? But it has no markers whatsoever of Alzheimer's, none whatsoever of the brain part. So is this not just a routine tauopathy, which we now know to be one of the most common variants of a frontotemporal dementia? And the answer is, it is a tauopathy, but it's not like any others. So this work really started from a unique piece of evidence at the bedside, which we then went on to look at the neuropathology. We then went on to do the isolation of the proteins. So at that point, my lab was starting to grow, and you could probably cut this in half and say, well, this is the group that was doing a lot of molecular work, trying to understand what was triggering the motor neuron degeneration, but then a whole group of us really trying to understand what was going on in the cognitive domain. About what point, Alex Mashinsky came to join me as a PhD student. And Alex really picked up from the work that Wencheng had been doing, as well as a colleague, Lou Gehrig, who came to do a degree with us as well. And what both of them, both May and Alex, were able to demonstrate was that if we took both non-neuronal cells and also neuronal cells, these are non-neuronal, these are HEC293 T cells, but if we transfected those cells with tau proteins, so the human tau proteins that were constructed in a manner to be either pseudo-phosphorylated, so they thought they were phosphorylated at the 175 site, or they were wild type, or we blocked phosphorylation there. What we saw, and the open bars here, are every time we transfected with a pseudo-phosphorylated 175, we got a dramatic upregulation in the formation of fibrils within the cells. This is normal, fine filamentous structures. This is what you should see. But this is what we were seeing even within 24 hours of constructs, and quite reproducibly, but also seeing an induction of cell death in association with them, telling us that these were likely pathological fibers, which was fascinating because we really needed to try and understand what was giving rise to that. And again, to make a very long story short, what Alex found was that the pathway was leading to an upregulation of GSK3 beta, particularly the phosphoisoform of it. And that phosphorylation was working into a cascade. So what was happening was, for some reason, 175 was being phosphorylated. That phosphorylation at 175 was leading to phosphorylation of GSK beta. That GSK beta phosphoisoform was actually then feeding back to the tau isoform and phosphorylating at a second site, 231. And it's that 231 which opened up that hairpin or that paperclip that we were talking about, and now opened its accessibility to start to aggregate. And so a very important pathway to be seen. But that's not enough. And particularly in our world, where people simply didn't want to believe that there was a dementia associated with ALS, and certainly did not want to believe that there was a tauopathy in association with it. And yet we were looking at pretty clear data, and I was going to meetings and presenting this, and colleagues were saying to me, Mike, there's got to be something wrong with your water or your assays or whatever you're doing, because we don't see this when we go looking for it. And so it's nice that you've got this pathology, it's nice that you've got a cell culture model, but you're going to have to show us that you can actually reproduce this in an animal model. And how do you do that? And I like this slide. I actually took this while we were traveling in the Galapagos. And I take it because, as you remember, from Darwin's period of time, right, it's a survival of the most adaptable. It's not, right? As It's not survival of the fittest, it's the most adaptable. And I love this one because these are all just crabs, right? They're all sitting on a rock in here. But, here's this outlier, right? Now, they're all the same crabs, but this guy's strikingly different. And we could wax eloquently for hours about why is he looking different? Well, I mean, is he as obvious on the rock as this one? If you were looking for a snack, you'd probably go for this guy as opposed to this one. So maybe it's an evolutionary pressure to become more like a rock in your coloration. I have no idea. But if you don't do that evolution, if you don't change things, you don't take a little bit of a risk in your experiments, you're not really going to move them forward. So we took a big risk. We went on to do something we'd never done before, but was described in the literature, so how hard can it be, right? Alex would shoot me right now if you heard me say that, because it turned out to be quite challenging, but it worked. And that was to say, could we actually do the same thing in an adult brain that we were doing in cell culture? And to do so, we worked on rats. And not mice, which is the traditional way we go for so many things, because rats don't express the full repertoire of tau proteins that we need to see. They have a smaller repertoire, but rats do. So mice don't, rats do. And so what we did was we actually did adenovirus somatic gene transfer. So basically what we took is our AAV9 viral vector. We modified its construct to express human tau protein. We took that tau protein and we did the same three things that we did in the cell culture. We either put wild type tau in, or we put tau where it was blocked from phosphorylation at 175, or we put it pseudophosphorylated in, and we put a GFP tag on it so we could track it. So the first thing is that with a GFP tag and then staining for it, AAV9 has a really interesting propensity for neurons. This shows you the deposition of our virus. Now, I love this slide for another reason. If you do any neuropathology, you're going to look and say, gee, Armstrong, what's that hole? Well, that hole is because we missed the hippocampus and we injected it here into the thalamic structures of a rat brain. And yet it's still migrated into the dentate gyrus, CA1, CA2 predominant domains. So we got transfection. And when we looked at the higher magnifications, we could show that the tau was being deposited, both the 175 as well as the wild type. We were getting some activation of GSK3 beta, and this was TDP43, evidence of a neural injury in there. So everything that we wanted to see happening, and this was at 1 month, but not enough time to really get a phenotype. So we did it for over a year. And after a year, we went back. We'd done all sorts of testing during this time period, and we looked to ask the question, were we getting tau pathology in the brain? And the answer is clearly yes. And we were getting not only what we call dystrophic neurites, so here we see some beating within those, we see the fibril-like formations, we even see neurofibrillary tangle-like formations forming within this. We didn't see these in the control, clearly suggesting that the 175 that we had discovered in the human brain works not only to produce pathological fibrils in the cell cultures, but also in an animal brain that expressed other tau isoforms. But then even beyond that, we were able to look and say that that 175 phosphorylation, the pseudophosphorylation, was driving phosphorylation of 231, and we could co-localize the two together. And remember what I said was that 175 drives phosphorylation of GSK3 beta, which then comes back and phosphorylates at 231, and it's the one that opens up. So that was really important. And then we also showed that it was inducing cell death. So everything that we had seen in cell culture, we were now able to demonstrate in an animal model. But in our world, still presenting this at meetings, that's nice. That's a rat. What has that got to do with ALS? Because we've never seen an ALS phenotype of any relevance in a rat. We use mice and such. But there had been a group that had discovered that we could actually create a genetically modified rat that would express mutant TDP43. It's one of the familial variants of ALS, and go on to get a motor phenotype. So we thought, great, let's just get the rat. And then we're going to do the AV9 experiment, which is wonderful, except that the investigators who had actually developed the rat had actually lost their funding and had to get rid of the rat colony and just keep a few embryos hanging around, which they then put into what's called the rat house in Kansas. So we had to start from scratch and get their embryos and rederive the entire lines. and ensure that they had a tetracycline responsive promoter, because as soon as you turn on this TDP43 gene in the mutant, the rats die within 12 days, which doesn't help us. So we went back and did the same experiment over again, except we did it now in rats that were expressing, neuronally expressing TDP43 in the mutant isoform. And we're developing a motor phenotype. So people would say, yeah, that looks like it might in fact be a ALS phenotype. And so we did that and did the AAV9 injection after 6 months into them. And sure enough, not only did we got the pathology again, thankfully, exactly what we wanted to see in terms of the neurofibrillary tangles, also dystrophic appearing neurites and such. But we saw that in those animals that had both the TDP43 expression, so had the phenotype of ALS and the AAV9, the degree of pathology within the neocortex was markedly upregulated, as it was within the spinal motor neurons as well, even though we had no evidence whatsoever of tau pathology in the motor neurons. So remember, early on, I said to you that the importance of the dementia in ALS was that it had a shorter disease course, that people were dying by a year earlier if they had both the frontotemporal dementia and the ALS phenotype in play. And this proves that it's a biological effect that was making things worse. Really important for us as we move things forward. Okay, so we're doing a lot of work right now to understand why that's going on. But I also say that, you know what, that's been a great pathway to figure that out, but there's other things in life. And this is an important piece as well for it. Your friendships that you make during the course of all of this, these people that I've worked with, the associates that I've had that I've met through my graduate studies, postdoctoral, through meetings, through being on peer review and such, have been the friends of a lifetime for that. And it's those things that really give you meaning to your life for that. And we have very close friends here. So this is actually chief of neurosurgery. While I was chief of neurology, this is Andy Perron and his wife Lisa. This is my wife Wendy, and we have this bucket list of trips that we're doing in our lifetime. So this happens to be on safari in the Serengeti, and a lot of fun as we've gone forward. Here we are in the Galapagos, standing at the edge of a volcano that had erupted literally about 7 years earlier than that. So people that you can share things with. But in the course of all of those, you meet people. And one of my favorite authors and people to read is Mahatma Gandhi. But this quote, listen as if you were to die tomorrow, learn as if you were to live forever. And I want to raise something that's really important. And it's been important to me, particularly in my career as a dean, and now very important to me in the role I'm at the CIHR. So this is a Maasai child in the Serengeti. And a lovely young child for that. But we had learned early on when we were there, because we visited a couple of medical clinics and such, that there was an interesting practice that when Maasai women became pregnant, they were immediately put onto bed rest. And they went on to a starvation diet. And they would lose weight, dramatically lose weight during all of this. And both Andy and I, we looked at all this and kind of said, wait a minute. Well, first off, in Western culture, that's just wrong. You don't do that. Intrauterine growth retardation is a major issue. It has all sorts of complications in association with it. And why would you do that? And it was very much like, okay, thank God we're here to tell you you're doing the wrong thing for it. And typical sort of, we know everything. Well, we don't know everything. These women and their children do exceptionally well. They come through without any difficulties for it. So it wasn't until the end of our trip when I had this opportunity to spend some time with a Maasai healer. And this individual was also working closely with a group out of Cambridge as they were starting to ask questions about how treatments in this area of the world were being advanced. And we sat down one night and I said, okay, why? I said, this makes no sense to us. And it was terrific. He said, listen, you come from a Western cultivation or civilization. You at a quaternary care hospital. And he said, I know how they work. If you have a child that's in distress, a breech, whatever it might be, a footling, you've got a helicopter, and that child and mom are on a helicopter and in your operating room within 30 minutes or so for that. So you have those. We're in the middle of nowhere. We're living on the plains. And if something goes wrong, we got nothing. And so we learned generations ago that when you do this type of a diet, yes, the mother loses weight, but the child is smaller. And so the complication rates during a regular vaginal delivery are dramatically less because the child is smaller. And then immediately afterwards, we put them on a goat's blood diet. And so we give them iron and we rebuild right away. And the growth curves for the child within a month are back to the growth curves of where they should be. So it's a historical event. What I learned from that is that we've got a lot that we can learn from other cultures about medicine, about how we practice, about the thinking that goes on for it. And it's really important for us to not assume that we really understand everything all the time. So it was kind of a bit of a humbling experience, but also one that I've kept with me for a long period of time. So what's next for me in all of this? Well, I would never, ever have guessed I'd be doing this in my life. Have the opportunity to be president of the CIHR. You know, we are the only federal funding agency for health research in this country. We have a mandate to do research that will change the health outcome of Canadians, and that's all Canadians. You know, and it's everything from our basic science work all the way through to understanding how we influence policy development through the research. And if you had asked me 30 years ago, when I was just finishing my postdoctoral work at the NIH, would I be doing this? Would I be giving you this talk today? The answer would have been, yeah, give your head a shake. This isn't going to happen at all. But it has. So I don't know what's coming up next for me. I'm loving what I'm doing right now, and I'll continue doing it. But this is a quote that I've had by my desk since I started university in '76. And I love this quote by Theodore Roosevelt. And it is, I am not smarter than the average man, but by George, I work harder at it than the average man. And I've kept that to this day. I still have it. So that's my career. That's my journey. And I'm hoping you've got a little bit about science, a little bit about maybe my philosophy of how I've gotten to where I am, a little bit about my life philosophy. And I'm happy to answer any questions you might have. I'll stop sharing the screen now.